Okay, let's do some recap question. I know I skipped them last time because they came in the middle of not finishing the lecture yet. So let's look over them. I added one more question that wasn't there before. So what are some examples of protein analysis categories and applications? So when we say protein analysis, what does that encompass? Yes, Lisa?
Quantification.
Huh?
Quantification.
Quantification is one. So in protein analysis, we quantitate protein. What else can we do? Sequencing, so protein structural analysis. That sequencing is one of it. So we're talking about all the bigger categories. So protein quantification, structural analysis, separation and purification, and then characterization of structure and also identification. They can go into one category. Okay. Application, there are many applications. We gave examples last time about needing to quantitate, needing to isolate, needing to characterize structure. Can we just talk about a couple examples? Lisa?
One for quantification would be Kelvin.
Yes. In what cases you would want to quantitate protein?
Oh, in what case?
Yeah. Mm-hmm.
Like nutrition.
Yeah, nutritional labeling, that's good.
If extraction is efficient.
Determine the efficiency of extraction by determining the yield of the protein.
And quality control.
Quality control, because protein content is really also important in terms of cost of the product and also in terms of the overall quality. So you want to make sure that your protein content is consistent throughout your processing and in your end product. Okay, other than quantitating protein, you're out of the category. In what cases you need to isolate and characterize the protein? Isolate.
To see the impact of processing.
To see the impact of processing, yes, okay, so we isolate the protein to determine if there has been any structural change due to processing, okay? But we also isolate the protein for a very important reason. For research, oh yes, okay, that's a very important research. Isolate the protein to determine its structure and functionality. But in industry, why do they isolate proteins? Huh? Ingredients. To formulate protein concentrates and isolates for different applications. Okay, so in terms of structural characterization, when is it important to look at structure? We talked about impact of processing, but can you give another example when you look at protein structure? We have a bunch of examples from last time that I talked about. Okay, review them. They are all written in your slides. So a bunch of examples looking at protein isolation methods, how different isolation methods might impact structure, how that might impact function, valorization, looking at byproducts that are high in protein, looking at the structure of the protein and see the value of it in terms of nutrition, in terms of functionality, changing processing equipments and looking at the impact of structure on functionality, looking at competitors in the market selling the same type of product, how they differ in structure and function. So there are so many different applications for doing that. Okay, what are some methods for protein quantification? We said Keldar is one. There are so many other methods. On what basis we choose the method, it really depends. Go ahead.
Or I was going to say money and time.
Money and time, and also food matrix, whether it is an isolate in a solution. You can use a research method that is based on measuring absorption and having a calibration curve. Or if you want to quantitate a complex matrix, you use the nitrogen-based methods, Kaldal and Dumas, nitrogen combustion method. So, and definitely time and cost is a big factor in choosing. On what basis protein separation protocols are developed?
Like what's all in the sample, so you decide like the order that you do it based on how you're trying to get to each component?
Yes, based on the composition and a matrix of the sample. Is it high in starch versus high in fat versus high in fat and fiber, starch and fiber? What do you have there? And also on what basis,
What you want the end product to be, whether you're looking for like an isolate or like 30% protein or 60 or.
exactly, what am I looking for in terms of purity? Am I looking for a very pure protein with a very bland flavor and maximum functionality? Or am I looking for a product that it's okay to have some fiber, it might help in extrusion, such as soy protein concentrate. It's 65% protein, but it has some fiber, and that could be good for different applications, such as extrusion application. But if I want to say specifically protocol, I mean, yeah, Yara.
Is the nitrogen? Did you look at the amount of nitrogen that you got in the sample? Most of the methods that you use is based on nitrogen.
So I'm looking for method that we choose to isolate. On what purpose we choose a method to isolate? On what, not purpose, but what, based on what I would choose to isolate.
The biochemical properties
Yes.
like solubility.
Exactly. I wasn't phrasing it right, the question. But yes, based on the biochemical properties of the proteins, so the differences in solubility, the differences in impact of pH and salt and solvent, what we know about the protein and what we know about its structure, its net charge, its percentage of hydrophobicity, then we choose the methods accordingly. What chromatographic methods we talked about last time that we can use for separation of proteins? Size exclusion chromatography is a very common one. What else?
Oh, I was going to say ion exchange. Also.
Ion exchange, huh?
Affinity.
Affinity chromatography, ion exchange. What did you say, Vaidi?
Ion
Oh, I thought you said hydrophobic interaction. Got it, fan. So ion exchange is actually applied in industry. We gave the example of Agropur, formerly known as Davisco.
Isoelectric precipitation.
Isoelectric precipitation, but it's not chromatographic method.
Yeah.
Yeah, but it is a method to isolate protein. You are right there. Yeah, I was giving you an example that ion exchange is actually used in industry to purify whey protein isolate. Okay, examples of protein separation approaches based on differences in solubility. Katie was ahead of the game answering isoelectric point, salt solubilization, heat, organic solvents, and heat, denaturation based on thermal treatment. Okay, we talked last time, the last bit we talked about were chemical assays for structural characterization, and we stopped there. So can you give an example of a chemical assay, and what do we look for in that chemical assay?
OPA,
OPA, what do we look for, Vaidi?
amino groups, free amino.
Free amine groups, and this we use to determine degree of hydrolysis. So OPA reagent react with primary amine. So the higher the degree of hydrolysis, that means the more you break peptide bonds, you're releasing free amines. So you can determine degree of hydrolysis based on the interaction of OPA with free amine. It's okay. And then OPA is also used to determine blockage of amine groups. So if we're looking at Maillard-induced glycation or Maillard reaction to determine blockage of lysine or loss of lysine, we can also measure OPA. Before processing and after processing, then we see the drop in amine group that react with OPA. Okay, OPA. What else? Okay.
Surface hydrophobicity
Surface hydrophobicity. So,
and surface
and surface charge. But they're two different assays. Surface hydrophobicity is really dependent on using a reagent that will bind to the hydrophobic surface of the protein, and usually it's the ANS reagent that you combine, and once it binds to the protein, then you measure fluorescence because it is a fluorescence compound that fluoresces. Surface charge, you can do that by a series of titrations, but you can also do that by an equipment, which we have a zeta sizer, for example. It can measure surface charge of the protein. So surface hydrophobicity, surface charge, amine groups. We have very important one, gel electrophoresis. It's a very common method to look at molecular weight distribution, any polymerization that can occur during processing. We see the band sticking at the top of the gel, not moving down because they're large. Different interactions, because you can solubilize your protein in different buffers, so you can elucidate what kind of interaction between the proteins. So if we solubilize in SDS and then load, we know that the interactions could have been non-covalent. If we put a reducing agent and then we see a change in the size of the subunits, we know that they were interacting via disulfide linkages because the reducing agent reduced disulfide linkages. So we can look at the different subunits within a protein ingredient as well. So it's used for many different reasons. Okay, any questions that you thought of? Yeah.
So the gel method that you're just talking about for SDSK, that the actual gel is technically like a gradient, right? So like if you had the 4 to 20%, does that have the whole? Like
The gel itself is a gradient. So it starts with the large pore sizes, and the pore sizes get smaller and smaller, because then you will have from 4 to 20% acrylamide. And the higher percentage of acrylamide, the smaller are your pore sizes. So you can run a gradient gel, you can purchase a gradient gel. When I was a student, I prepared my gradient gel, and it was I'm not gonna swear, a very tedious procedure to prepare a gradient gel. Takes the whole day. But then you can buy them, or you can buy one concentration of a gel, acrylamide.
So what is technically Western blot? Does that use like?
Western blot is it's a blot. You blot. You do the gel first, and then you transfer the proteins on a membrane, and that's the procedure. And what you use in terms of solvents and all that is Western blot. There are different types of blots. I think there's the Eastern blot too. I really don't know the Eastern blot. I know Western blot. We do it in the lab. But the Western blot, you start with running a regular gel, and sometimes it's a 2D gel, two-dimensional, running by isoelectric focusing and then by size exclusion. And then you transfer into a PVDF membrane. I will talk about that a little bit today, the Western blot procedure. Yeah. Okay. Any other questions about previous lecture? I mean, don't hesitate to come up with questions as you study and review. It's really time to discuss, not just for participation, just to clarify anything that you'd like me to explain more.
Explain the capillary electrophoresis.
The capillary electrophoresis. Okay, so really I went over it very briefly because in this class we really don't spend a lot of time talking about principle and methodology, but if you go to the food analysis book, you can read about it more, but it's based on the principle of gel electrophoresis in terms of separation of the protein. So you can separate based on size by having an acrylamide gel inside a capillary tube, and then the proteins will be solubilized in SDS. So they all will carry a negative charge, and they will separate based on size. The difference here is as the protein separates, they go and be detected like an HPLC detector. So that's the beauty of a capillary electrophoresis. You don't only separate based on size or charge. You also can detect or quantitate as well. So you can do quantitative, not just qualitative like a gel, right? And then you can have the isoelectric focusing so that you will have all of the ampholytes that you run and form a gradient of a pH gradient, and the protein, once it reaches its isoelectric point, it will sediment. So they will separate based on isoelectric point. And then you have the capillary zone, which is negatively charged selenol groups, and the positive charges from the buffer that go towards the cathode will bind to the negatively charged, and they will have a pull for all of the charged proteins. The positive charge is first, negative, and then the neutral will be separated. So that is in brief the description, but if you really want to learn more about it, you have to go back to the source. Okay? Okay. Anything else? Okay, always think of questions and use the first 10 minutes of lecture to just for discussion time. Okay, so I'll spend some time now talking about instruments that we used or analytical tools that are instrument-based that we use for structural characterization. Specifically structural characterization, and in other cases also identification of the peptide or the protein. So if we look at structural characterization using instrument, the very first one that is the most common for looking at protein denaturation is differential scanning colorimetry. Are you familiar with it? Who doesn't know what a differential scanning colorimetry is? And it's okay if you don't know. Perfect. So what we do in this instrument is you put your sample in a small compartment, container, and then you place that in a compartment in the instrument, and then you generate a heat flow within the instrument. You program it to increase the temperature over time. Sometimes it's 5 minutes per minute, or 5 degrees Celsius per minute, or you can say 10 degrees Celsius per minute. It really depends on your protocol and on the separation of the peaks that you want to see. And the peaks that you see are not chromatographic peaks, they are called endothermic peaks. So these peaks that you see here in the thermogram, this is called a thermogram, are endothermic peaks. So this is a thermogram, and this is an endothermic peak. So as you increase the temperature over time, you get to a point where you provided enough energy for the protein to break few bonds that will increase the entropy so much and cause a change in enthalpy. So the area under the peak is the difference in enthalpy. So that's the heat uptake that resulted in unfolding or the denaturation of the protein. So how much heat the protein took to cause a denaturation and change in structure will be recorded as an endothermic peak. And the area under the curve, again, is the delta H, which is the change in enthalpy. Okay, so here's an example where we have used it. It's been published from a journal article. We looked at a commercial soy protein isolate and a soy protein isolate we prepared in the lab. And we were looking at the functionality of these proteins and the selectivity of certain enzymes on hydrolyzing these proteins and subunits within these proteins. So looking at soy protein isolate commercial, we didn't find any endothermic peak. What does that mean? Can you guess? Rachel's gonna guess. I want Becky to guess.
The protein was denatured?
Yes, thank you. I know you know it. I just wanted to hear others. Okay, yes, the protein is already denatured. There is, no matter how much heat you're giving, there is no detectable change. So it is denatured. Whereas in soy protein isolate we prepared in the lab was prepared in a way with minimal thermal treatment. So we were still able to see two endothermic peaks for the two main subunits within the protein. The beta-conglycinin, we'll learn about them later, and glycinin. The one that has higher denaturation temperature is the protein that has disulfide linkages in it. So with disulfide linkage, you need higher energy to denature the protein. So one denatures at onset of denaturation or denaturation temperature. There's the onset at the beginning of the peak, and then the middle of the peak is the actual denaturation temperature. And it is 75 degrees for one protein and 92 degrees for the other protein. And the change in enthalpy is the amount of energy required to denature that protein. And it is corrected for the amount of protein, so it's joules per grams of protein. Okay, and then you see here that there is another one that we looked at, which is a purified fraction that is just glycinin. So we took the SPI and fractionated glycinin, and we were able to isolate it and determine its denaturation temperature, which was obviously similar as the glycinin within the complex matrix. And we did the same for beta-conglycinin. We took it out of SPI and measured it separately, and we were able to get the exact temperature of denaturation. Okay, it's a very cool unit when it functions.
So the order of the lines as you go up, does that have any?
Oh no, this is just, we overlaid them, but you run them separately as a sample. These, for the sake of publication, we just put them in one. And this is our kind of guide here, that the heat flow was 0.1 heat flow units, milliwatts. Okay.
Why would the glycinin subunit have such a higher enthalpy than the glycinin in the soy protein prepared?
Here and here?
No, like for the same glycinin subunit, why is it so much bigger for like the pure glycinin versus the glycinin in the soy?
That's a very good question. I didn't think anybody was going to ask that. But it's a great observation. When we do that, here you have in 1 gram of protein, you have a mixture in terms of ratio, how much of this you have versus this. But here, the 20% protein solution that you have is mostly glycinin. And this is directly proportional to the amount of protein you have in this. So it looks bigger because there's no other peak there, no other protein. Okay. Cool. Okay?
Is there any interaction between two components in the system to affect the entropy?
Yes, there are interactions between the proteins that can impact their denaturation, and not just that, it's not a system in void. There are the salts and sugars that will also impact denaturation, and also the moisture content.
Okay, so in this graph, it's not only the pure glycinin have the higher, because it's like concentrate. It might have like interaction between that, so it affects. I don't know.
Here, although we say it's glycinin, it's purified fraction of glycinin, but there are other components. So it's not 100% glycinin itself. So there are other components, and it really gets complex in the protein world to determine what component exactly is contributing to the stability or the instability of the protein. There are so many different components that can play a role. One is the moisture content. So if we run the same DSC denaturation at different moisture content, we're going to get a different outcome. We're going to get a different outcome in terms of temperature denaturation and enthalpy. Okay? And if you have salt or sugar, also you're going to get a different outcome. We're going to talk about salt impact, sugar impact, moisture impact in the next lecture.
Would
Would you ever get a negative enthalpy?
Oh yeah, well this is depending on the program. It's not really negative, it has to do with the program. Sometimes it is a negative peak telling you the uptake or the energy needed to break versus energy of uptake.
Okay.
It depends how it is programmed, if it's measuring the intake or the output, input or output of energy. So sometimes you have your thermogram is in the negative, but the delta delta H should be the same, whether it's reading negative or positive.
Okay.
Because your delta H is a change in enthalpy, whether not a positive or negative number. Huh.
So I've seen some where they don't get a good baseline, like
Yeah.
it like starts to just go up. What does that tell you?
Something is off with how you're programming.
Oh.
Yeah, and sometimes you have to change the rate of increase in temperature to get, if you have two proteins that are very close to each other, so you slow the rate or increase the rate, and they have to play with the program to get good separation. And it really depends on the instrument itself. Yeah. And sometimes if there is a leak too, so if you're heating and then the small container bursts, then you have variation in baseline as well. Jason, you had your hand up?
No.
No? Okay, great. All good questions. Keep them coming and we will never finish the material. You'll have less to study. So that's okay. I'm happy to eliminate material as long as you study, so you understand the material I'm covering. It's fine. Okay. FTIR is another powerful tool that we use in protein structural characterization. And again, I spent a whole lecture or two in IR in a different class, so here is not the place where I go over principle very in detail, but I'll give you a very brief overview. In Fourier transfer infrared spectroscopy, what happens is different functional groups that is composed of different types of bonding will absorb IR radiation, and then you will have different, it would be due to bending or stretching of the bonds. So you will have an increase in vibrational energy once IR radiation is absorbed. And that is the absorption of the IR radiation is detected and quantitated and related to certain functional groups within the protein. That is my 1 hour lecture in analysis in 2 minutes here. So in terms of application, here's an example where we looked at FTIR to look at the secondary structure of the protein in flour and then in dough system. If you think of a dough and you want to understand what stabilizes a dough versus another type of dough, so one dough is made up of wheat flour, another dough is made up of corn flour. The dough from wheat is more stable because of different reasons. One reason is the formation of beta sheet. Beta sheet is more stable in the dough. When it rises, it stays more stable, and when you bake the bread, it keeps the bread from collapsing, the beta sheet structure of the protein matrix. Whereas in corn flour, for example, the proteins don't form beta sheet, they form more alpha helixes and random coil. So that's why a corn bread collapses if you don't add an ingredient that stabilizes the dough matrix. So here we look, mainly when we run FTIR, we are really concerned with looking at the secondary structure, looking at the ratios of alpha helixes versus beta sheet versus random coil versus beta turn. So these are, and it is the amide 1 region is where there are multiple regions. It's called amide 1. I'm not going to go through details there. But the amide 1 region, if you deconvolute that peak into individual frequencies, you will get the beta sheet frequency, the unordered or random coil frequency, the alpha helix, and the beta turn. So we can quantitate the area under the deconvoluted peaks and determine the relative abundance of the different structures or secondary structures and how they change upon processing, physical kneading of the dough versus after fermentation of the dough, and or any other application if you're let's say extracting a protein from a meal using different methods of extraction, and you want to determine denaturation at the secondary level structure of the protein. You can look at, for example, the ratio of alpha helix to beta sheet and how does it change during processing. Okay. So here in this publication, we looked at the flour and the dough. And the dough, you can see this huge peak. This is the water peak. Water is a very strong absorbent of IR. So if you're looking at anything in this region, you really need to correct for the water peak. But we don't care about this peak because what we're caring for is the amide 1 region and the secondary structure of the protein. So in this example, we looked at differences in secondary structure of the protein in a dough made up from 100% wheat versus addition of a different grain, which is intermediate wheatgrass, that is related to wheat but doesn't have the same function. So here 0% intermediate wheatgrass, that means 100% wheat, and this is 50% inclusion, 75, and then 100% intermediate wheatgrass. And the difference, we looked at beta sheet. The more we add intermediate wheatgrass, we have less beta sheet, which explains less stability of the dough. And the more we have intermediate wheatgrass, we have more random coil. So less beta sheet, more random coil. So that gives us an explanation of why that particular grain behaves differently in dough stability and strength. Okay. Another very powerful tool of structural characterization is Raman spectroscopy. It is complementary to FTIR, and here you're not measuring the absorbed light or the absorbed IR light, you are measuring the scattered light. So in Raman spectroscopy, what happens is when you project IR radiation onto your sample, it will absorb it, and then as it returns to the, returns to the ground state, it would scatter the light. The light scattered is of less energy because it not always returns to the ground state. Sometimes it returns to a little bit higher energy state. So the light that is scattered, that is of a lower energy than the light that hit the sample, is the Raman scattering. That's what it is. Again, there is an entire section to read about in the food analysis book, for example, if we want to learn more about Raman. But it's basically the light scattered after excitation going back to the ground state. So that light scattered is very characteristic, and sometimes it's really very weak. So this is whey protein, for example, beta-lactoglobulin. If we just put it on a slide and then project the light, the IR radiation, and then look at the intensity of the light scattered, it's really very small. So it's a very, very insensitive tool. However, we can make it more sensitive by converting it to a surface-enhanced Raman spectroscopy. The surface-enhanced Raman spectroscopy, so this is a scanning electron microscopy image of silver dendrites. Silver dendrites is a ruffled surface where the protein can bind to. And when it binds to it and then project light on it, IR radiation, you have a very good signal, much enhanced signal. That's why it's called surface enhanced Raman spectroscopy. And an example of application, this is where you spot your samples. You have the silver dendrites and you spot your samples on it and you project the light. This is an example application. We used Raman spectroscopy in it. And here, what we were looking at functional groups changes to explain changes in structure and functionality of whey protein isolate once we added a sugar to it. This is partially glycated whey protein. So we linked carbohydrate moiety on the protein, and we saw that the solubility of the protein improved. So, and we wanted to look at what happened to the structure that resulted in an improvement in solubility. So, for example, we ran it on Raman, and we saw shifts in peaks, and a new peak formed. A new peak we saw at 983, and when we looked at literature, that was the confirmation that we got a carbonyl group reacting with an amino group. So that is your Maillard reaction bond. So we were able to confirm that this actually happened because we didn't see it in whey protein isolate, but we saw it in the glycated sample. We also saw shifts in disulfide linkages due to changes or intermolecular changes. We call it disulfide interchange. So we saw some shifts in that. We saw shifts in tryptophan and tyrosine that indicated that the protein opened up partially. And one important change, and two important changes, is we saw change in beta sheet. And beta sheet increased so that we have more thermal stability. So with glycating, the protein was soluble under heat treatment at different pHs, and that's because we have more beta sheet in the structure. Also, the carboxyl group in the protonated form was not present, or we were not able to detect it in the glycated. That means it's mostly in the ionized form. And when it's ionized, it's more soluble, obviously, when you have more charges on the protein. So, using the Raman spectroscopy, we were able to look at a lot of different functional groups and how they change, which explain why we saw improvement in functionality, enhancement in thermal stability, increase in thermal denaturation temperature. So the change in structure, we were able to relate that to changes in functionality.
Does the area under the curve mean anything?
Huh?
Does area under the curve mean anything in Raman, or?
Yes, it means the intensity, because you measure intensity is area under the curve, and you measure shifts in wave number as well. Yeah. So for example, here the SS linkage is shifted from 508 to 542. It didn't have a different position of SS linkages occurred. So you see a shift, and then an area under the curve is the intensity. Yeah.
Could you go back to this blotting thing that you did with under the yes? Can you explain that again? Why would you do that?
Why would I do this, or why would I do that?
That.
This? That's
Yeah,
your sample slide. So you will prepare your sample here, you have your silver dendrites, and then your protein will bind to the silver dendrite, and then you take a sample from there and you spot it. And then usually you run the scanning on multiple spots, and then you do PCA analysis.
Okay.
Okay. the PCA analysis you do to determine which of the changes in the shifting of the bands and the intensity resulted the most in a structural change. That's PCA, because you have so many different functional groups, you would see which ones represented the most change. You have so many of them. Okay. Another important tool for structure characterization is size exclusion, HPLC combined with MALDI, multi-angle laser light scattering. So, which gives you exact molecular weight. And then what you do here is you look at not only the molecular weight of your individual proteins, but if you hydrolyze, for example, you can tell which subunits was hydrolyzed and what is the range of the molecular weight of the peptides you've generated. You can also look at aggregation. So if your protein was subjected to processing that resulted in dimer formation or trimer or a large polymer, so you can also look at the changes in the molecular weight. And then you can solubilize your sample in different solutions to determine whether interactions between the proteins were covalent or non-covalent interactions. So here's another application for size exclusion chromatography that we have done in the lab to look at proteins. Again, the example of intermediate wheatgrass versus wheat. And then we look at the presence of the high molecular weight polymeric proteins that are the glutamins, mostly responsible for the dough strength and elasticity. And then you look at gliadins and then albumins and globulins. And we have the SDS extractable or soluble protein versus insoluble proteins. We got the soluble proteins and we got the insoluble proteins, and we looked at changes in ratio of the different distribution. Because if you remember from FTIR, I said we had less beta sheet, more random coil, and less stability of the dough and strength of the dough. It has to do with the composition of the proteins. So intermediate wheatgrass, based on size exclusion and then LCMS later on, found that we have more of the gliadins and albumins and globulins and less of the high molecular weight polymeric proteins. This is another example. We use size exclusion. This is Chelsea's work. What we were looking at is hydrolyzing with different proteins or different enzymes. So the first one here, A, is whey protein isolate. Don't worry about B. Whey protein isolate or concentrate subjected to protease M, one type of protease. We can see that peak 5 from running a standard curve. I don't know why it's not showing very well. It's alpha-lactalbumin. And peak 4 is beta-lactoglobulin dimer. Because depending on the pH, especially pH 7, it will form a dimer. So peak 4 is beta-lactoglobulin dimer, peak 5 is alpha-lactalbumin, peak 3 is lactoferrin, peak 2 is immunoglobulin, and peak 1 is insoluble polymer, just a large polymer. So we were able to determine that both from literature, knowing the molecular weights of the different components of whey protein, and also running a standard curve, running standards on the column and plotting the molecular weight of these standards so we can deduce the molecular weight of these peaks. And also, what was very important is that if you look at the figure C here, you see a decrease in the peak 5, fraction 5 or peak 5. There is a decrease compared to the first one. This means that the enzyme we used here is more specific to alpha-lactalbumin. It really hydrolyzes alpha-lactalbumin versus beta-lactoglobulin. and resulted in smaller peptides that we were able to detect. And then here, when we increased the hydrolysis time, I believe, we were able to get more of the alpha-lactalbumin hydrolyzed and more of the smaller peptides generated compared to the first one. And then here, we used different enzyme, it's trypsin enzyme, and the trypsin enzyme left alpha-lactalbumin intact, that peak 5 is still there, but peak 4, which is beta-lactoglobulin, was barely there anymore. So this enzyme was more specific to beta-lactoglobulin. So that was, and then more of the smaller peptides, like you can see here, you have more peptides generated here that are of smaller molecular weights. Okay. Chemical deviation, and sometimes it's not necessarily deviation, it's not here, but we can do identification. But since we're just talking about structural characterization, changes in structure, I'm talking about changes in the structure of the protein, for example, upon hydrolysis. Okay, upon hydrolysis or upon glycation, that means bonding of a carbohydrate moiety to the protein. But both MALDI-TOF and LC-MS-MS can be used to identify a particular protein or the origin of the protein. But in this case, my examples are for hydrolysis or glycation and identification of the peptides, the source of the peptides, the molecular weight of the peptide, and also the origin, where did they come from what subunits. And I'll give you research examples for both. Okay. MALDI, again, this is not the lecture of describing MS, but MALDI-TOF stands for matrix-assisted laser dissociation ionization time of flight. It's a mouthful. What it means, matrix-assisted, that means we have a matrix, a specific matrix, and in this case it's weak organic acid, where we mix it with our sample. And then what happens is we project a laser on the spot where we have our sample with the matrix. This is very highly active absorbent of light. So it will absorb the energy and heat up and causes ionization of the sample, either deprotonation or protonation. That means either giving it a proton to make it positively charged or taking out a proton to make it negatively charged. And then a voltage is applied where then the ions are accelerated down a flight tube, time of flight. So they travel based on their size. So the smaller ones will travel the fastest and reach the analyzer the fastest. So the smaller ones will actually come out first and be measured and converted to molecular weight. And the largest ones will have a slower rate of travel and will be determined at a later time, reach the detector at a later time. That's why it's called time of flight, the time it takes to fly into the flight tube. So again, this is a very brief and simple description of MALDI, but in terms of application, what we often do, especially for proteins that we want to identify, or if we want to identify a change at a particular site on the protein, you run the protein normally on a 2D gel, so we have as much separation as we can. We take the spot of interest, we hydrolyze with trypsin, and we obtain, the reason we hydrolyze with trypsin is a lot of the protein database for identification is based on trypsin hydrolysis, so the database of the peptides are based on trypsin hydrolysis, so a lot of that, a lot of the hydrolysis is done by trypsin. And then sample preparation, and then the samples are desalted and spotted on a MALDI plate, and then you can determine the molecular weight of the peptides that are generated. And from there, you can go back and ID those peptides. A common application is ladder sequencing. So if we want to sequence the peptides, you can do that by MALDI-TOF, by running cycles with an exoprotease. An exoprotease will break one amino acid at a time. So you can actually run it at the beginning of the cycle, and after one cycle, you see, for example, you get another peptide that is less by one alanine group. And then you run another cycle and you get a peptide that is less in molecular weight by one glutamic acid group, and so on. And then you can then ID your peptide by ladder sequencing. So here's an application that we use in our lab for MALDI. And again, back to the glycated protein, we wanted to determine where was the site of modification. So there are multiple lysines, so you can see here there are multiple L's, multiple lysines on the protein. We wanted to determine at which lysine position where you have primary amine, you get the carbohydrate to react via Maillard reaction. So what we did, we took the sample, we glycated it with maltodextrin or dextran, and then we subjected the sample first to alpha amylase hydrolysis to break the dextran groups, otherwise it will be too big to fly in the time of flight. So we hydrolyzed the carbohydrate first, and then we subjected it to trypsin hydrolysis, and we ran it, and we found a couple of unique peptides. This first one was present in the glycated sample, but it was not present in the regular sample, and it was 5 units of glucose higher than the original peptide. So from there we determined the peptide based on the library of trypsin hydrolysis, and we found out that the lysine at that position was actually the lysine that got that carbohydrate moiety attached to it. And why was that significant? Because we were able to determine the location in the protein where the modification happened. And that location was very critical for the unfolding of the protein. The presence of a carbohydrate moiety in that location prevented the unfolding of the protein at a particular pH. This is for a later time. When I talk about modification, I will talk about that location again. So the location was very important to explain why the protein resisted denaturation at the isoelectric point. Okay, LC-MS for also structural changes has often been used to determine selectivity of the enzymes, so what kind of peptides are produced, and the origin of the peptides, what subunits within the protein the enzyme was able to hydrolyze. So it's used to determine which protein got digested. It's useful in fermentation studies to determine the efficiency of protein digestion by certain enzymes during fermentation, or identifying peptides that have bioactivity, for example, and that is an application we used in our lab. And in this experiment, what we were trying to establish is generate a bioactive hydrolysate. In literature, when we were looking at literature before we started this work, we found that soy protein component beta-conglycinin contain peptides, sequences of peptides that are bioactive with antihypertensive activity. And we saw that a lot of people, to produce nutraceuticals, they will go and fractionate beta-conglycinin from soy protein isolate and then hydrolyze it to produce a bioactive peptide. And this is very tedious and not industry feasible. It's like, why do we want to go fractionate soy protein and then from the soy protein go and fractionate beta-conglycin? That's a lot of work and it's expensive. So we thought about, can we target beta-conglycin specifically with SPI without fractionation? And can we target the other subunit again without having to fractionate? So we ran the experiment by selecting an enzyme papain and hydrolyzed the soy protein isolate, and then fractionated those subunits and used papain to hydrolyze and pepsin to hydrolyze glycine. From previous studies, we know that papain can specifically hydrolyze beta-conglycin, and pepsin can specifically hydrolyze glycine. So here's our tests where using papain, we were able to get rid of beta-conglycin specifically, and using pepsin, we got rid of the glycine. So we kind of proved that using this SDS paper. And then we determined the IC50, or basically we're looking at antihypertensive activity in vitro. IC50 is the concentration of the peptides that result in 50% inhibition of an enzyme that is responsible for hypertension in our body. So the smaller the number, the better is the potency of inhibition. Okay? You would need less to reach 50% inhibition. So we found out, like literature, is the hydrolysis of beta-conglycin gives us more potency in hypertension inhibition than hydrolyzing glycine. Long story, we wanted to confirm that the peptides that were generated here and here are the same, and the peptides that are generated here and here are the same, to prove that we don't really need to fractionate. So we did LCMS, and okay, big table, but basically we looked at peptides that were common in both fraction, the pepsin hydrolyzed fraction and the pepsin hydrolyzed glycine enriched fraction, and the same for papain. And we found an overlap in the peptides that were generated, whether we had the entire soy protein isolate or whether we hydrolyzed the fraction of it. So we kind of proved that you don't need to fractionate a protein component off of the entire mix of proteins. But if we use an enzyme with a very highly specific activity, we can generate and release the peptides of interest without further fractionation. Okay. So, lastly, for structural characterization, you can use NMR to look to determine the configuration of a protein. And this is my least favorite. I don't know much about it. But basically, you can do proton NMR and look at, identify pairs of protons in close proximity to each other and do a puzzle determination of the spectra of NMR and then come up with the configuration of protein. I never did that. I don't want to do that, but it can be done. Okay. I have a few more slides. I'll go over those and then give you a break. And then we can do the questions together of the practice question. Okay, other than structural characterization, there is the identification of the protein. Identification tools as well. This can be based on composition and sequencing of the protein. So we can do amino acid analysis. And why do we care about amino acid analysis? Because we can determine nutritional quality by determining essential amino acid and finding out the rate limiting, or not rate limiting, the most limiting amino acid. That means the essential amino acid present in the lowest concentration. And it is needed for determining the PDCAS, protein digestibility corrected amino acid score, which we need the amino acid score to determine the PDCAS value. And identify a newly isolated protein. So if there is a new protein that we are interested in, like pennycress protein, it would be good to do amino acid composition to look at its essential amino acid composition, its percentage of hydrophobic amino acid residues, percentage of charged amino acid residues. So looking at characterizing it from the amino acid perspective. And sometimes you can determine amino acid and then deduce the molecular weight. By determining the composition of amino acid, you can calculate the molecular weight, which is the most tedious way of doing it. You better use LCMS to do that, or MALDI. Okay, how do we determine amino acid composition? So basically, the most common way is you hydrolyze your protein with a strong acid, 6 normal HCl for 24 hours with heat. And then you measure, you break all the peptides bonds, obviously, and then you measure the amino acid content by running it through chromatography, ion exchange chromatography, or reverse phase. If you do ion exchange chromatography and you have the electrochemical detector, you don't need to react it with anything, react the amino acid with anything. But if you do reverse phase liquid chromatography, you have a UV detector, you better have, you react the amino acid with an anhydride, and then you measure absorption. Or you reacted with ophthaldehyde and then you measure absorption or fluorescence. So you can do either. But not all amino acids are chromophores. Only 3 of them are. So you need to react them with something if you're using an absorption or fluorescence method of detection. And we already talked about using OPH, German pH, so I'm not going to repeat that. But sometimes, not sometimes, always, when you hydrolyze with strong acid, you lose tryptophan amino acid. So if you want to measure amino acid tryptophan specifically, we hydrolyze the sample with NaOH, strong NaOH, and then determine tryptophan composition. And terminal sequencing, which is used to determine the sequence of amino acid within a protein. And it's often called Edman sequencing because the guy that discovered it or came up with it, his last name is Edman. So what it's based on, first of all, you need a purified protein, as pure as possible, because it requires high purity of that subunit or that protein to determine the exact sequence. If you have a mixture of protein, then you won't be able to detect the correct sequence of your protein of interest. So you reacted with PITC, a compound, the coupling compound, that will react with the first amino acid from the N-terminal and break it off the chain, and then another chain of reactions will result in a compound that will absorb light at 270. So this is one cycle. Every cycle, you send it to reverse phase chromatography and you run standards so that you can quantitate that particular amino acid based on its interaction with the column. It determines where it comes out. So you run standards with that and run calibration curves. So I said it needs to be purified. So oftentimes you run 2D gel and you transfer the protein into PVDF membrane, and then you cut off the protein that you are interested in and solubilize it, and then run your conversion cycle, and and then run the HPLC. So identification can be done with ELISA and Western blot here to determine the presence of an allergen, for example, or residual allergen or protein that's not supposed to be there. For example, do we have horse protein in the beef patty? So you can run an immunoassay that will be looking, let's say, specifically for presence of horse protein. So you would have antibodies that are specific for that target protein, and then you will have an antibody that is labeled and specific for the antibody to the target protein, and it's labeled with an enzyme, and then you have a substrate that will be specific, the enzyme is specific to that substrate, and you generate a color and you measure absorption. That is, again, a whole lecture in another course that we talk about enzyme linked immunosorbent assay, ELISA. And it's semi-quantitative. You can run standards. You can actually quantitate, not only detect the presence, but quantitate. So if you have your target protein, the antibody will bind to it. The antibody to this antibody will bind, and it's labeled with the enzyme. Then you get a color. If you don't have the target protein, you won't get all of this chain reaction happening. In Western blot, it's more qualitative, and you're looking for the presence or absence of a certain protein. And if you're modifying, you want to see what happens to the immunoreactivity if you modify the protein. Would it still be detected by an antibody, or won't it be detected by an antibody? In Western blot, you run a gel, and oftentimes it's a 2D gel, where you get maximum separation of your proteins, and then you blot. That means you transfer the protein onto a PDF membrane. So there is chamber for transfer. I forgot to bring it with me, but you put the protein, the gel, sandwich it with the membrane, and then you put it in a compartment where you run voltage, and the protein will go in one direction and transfer onto the membrane. And when you have your protein on the membrane, you will wash with primary antibody. That means the antibody specific to the protein of interest you're searching for. So it will bind with it. And then we put a secondary antibody that is labeled, will be specific for that primary antibody, so it will bind to it. And then you incubate with the substrate, and then the substrate gets converted to a product with the enzyme, and then you see a color change. If you see that spot over there, you don't see any other spots, obviously. You won't see them. This is just for the sake of the image. If you see that one spot, you know that you have that protein of interest is there in your sample, and it's immunoreactive. So here's an example application where we looked at modifying soy protein to reduce its allergenicity by hydrolysis and then by glycation. So we subjected the protein to different levels of hydrolysis, and we transferred it into a membrane, and we did the immunoassay by incubating it with the antibodies. And we found that as the degree of hydrolysis increases, obviously we have less immunoreactivity. We see less intense bands here. And then when we glycated under different conditions of glycation, we also saw that at a particular condition, we saw an increase in intensity. That means the protein did not bind as much with the antibody, so that means we reduced the allergenicity of the protein. Well, that's an application for Western blot. Finally here just to illustrate that oftentimes when you use chromatography on protein, you use, to purify, you use several chromatography techniques in sequence. So in here, in this example, we were looking to isolate a bioactive peptide, in this case antioxidant peptide, from a mixture of peptides in a hydrolyzed sample. So we started off with ultrafiltration. What you do here is you separate based on size. So you get rid of anything, let's say, that is greater than 5 kilodalton. All the peptides that are smaller than 5 kilodalton will pass through the filtration and you collect that. And from there you run ion exchange followed by size exclusion, or size exclusion followed by ion exchange, it doesn't matter. But for first, let's say we did size exclusion, it doesn't matter. Then you separated the protein, the peptides based on size. You take each fraction and you run the antioxidant activity assay, and you found that fraction F has the highest antioxidant activity. You take F and you run it on a size ion exchange chromatography, and again it separates based on molecular charge. So, and then you run the assay, the antioxidant activity assay on each fraction. Then you take the fraction with the highest activity. And then you take that and you separate it by reverse phase chromatography, for example. That separates based on polarity. And then again, you measure the fraction with the different fractions, and you take the fraction with the highest activity. And then you get a purified peptide that you can sequence using either N-terminal sequencing or MALDI. You can determine its molecular weight. So it would be the most potent peptide in your entire mixture of hydrolysate. So this is to illustrate that if you want to get a very pure protein, a very pure peptide, you have to go through a series of different modes of separation. Because multiple peptides might have the same size, multiple peptides might have the same charge, might be very similar in polarity. So you do several levels of separation. Often you start with ultrafiltration because if you know the size you're looking for or the range of size you're looking for, you can eliminate a lot of proteins and peptides you don't need or you're not interested in by simply filtering your sample. Okay. All right, let's take 5 minutes break and then we can work on the practice problems together. So if you are back, just divide into two groups. And let's see here what we have. Okay. All right. Let's get started with working on your practice problems. So question 1 is still talking about structure a little bit. And then the other question for protein separation analysis and characterization. Let's focus on this. The first one does not require a lot of really discussion. You can get that from the notes. Unless you have a specific question, we can look at it later together. So I will skip that. And then I want each group to work on all of the components, but I'm not going to divide it into two. So A, B, C, D, and E. So we have 5 components. Let's discuss as a group, and then we can have one group answer A and B, and another group C and D, and then E, whoever. So let's take 10 minutes doing that.
Let me know. They would be,
Yeah.
Hmm?
Are you posting answer keys for these?
Hmm. So after next week, when you turn them in, I will post both keys.
Oh, yeah, but I because it's LSTS is guys. Yes, right. So LSTS, everything is like. The other one that we don't modify charge. It's just the page one, so it goes to next. It's right down. Yeah.
Are you ready to discuss?
Just A through E, right?
Huh?
A through E or?
Yeah, A through E. Are you guys, this group ready? This group ready? Okay, let's start with this group. Here, can you do A and B?
Us?
Yes, you. Who gonna? I would like to hear Lucy's voice
Okay.
because I don't hear it very often.
So for A, we said the salt ions will shield the charges on the protein and compete with the protein for water, causing the protein to aggregate by hydrophobic interactions and precipitate in solution.
Perfect. Did you guys hear that? The other group? Do you agree with that? Okay. Do you have any questions about that? Okay, good. B.
Okay, and then B B, we said you need to remove the salt to reduce the shield on the protein's charge, so that it'll actually want to interact with the column. So you can use dialysis or an ultrafiltration membrane to remove the salts. And then we just described dialysis, saying that it works by having the protein solution in a semi-permeable membrane that will allow salts to exit, but will contain the protein. And salt will continue to move out of the membrane until an equilibrium is reached with the surrounding wash water, at which point the wash water can be changed to continue to remove more salt by reestablishing the gradient.
Yeah, and you continue doing that until you reach a very small infinitesimal amount of salt. Yeah. And then if you want to describe the ultrafiltration unit, which I didn't have in my key here, what would you say, Rachel? Do you have that described?
I only did one of them. I thought that we will.
Oh, you only need one, but if you want to describe the other one, did the other group describe? Which one did you guys describe? Dialysis? Okay, but if you want, okay, I also have my key dialysis too, but because we talked about ultrafiltration in the class as well, so we will have membranes, also of semi-permeable membranes with different pore sizes that we select to be as small as possible to just allow salt to be removed, not the proteins to be lost. And usually you apply pressure by pump, you generate a flow through the membrane, and then your sample will go through the membrane, and the large proteins will be on the retentate side, and then the salt will be in the as permeate, and then you run several volumes of water through to wash as much of the salt out as possible. Sometimes you have to do 5-6 washes, and usually we run ash content to determine if we were able to get most of the salt out as well. Okay. The second group, you want to tell me your answer for C?
Okay. So we said that we would run the sample on the machine using a pH in between 4.5 and 6. And so at this pH, our target sample would have a negative charge. So then we would use cation exchange, which is a positive charge on the stationary ZAR protein. Anion.
Yeah, that is a very common, confusion. It is positively charged, but you're exchanging anions. Yeah, so it's called anion. Yeah.
So positive on the stationary.
Yeah.
Our target protein would then interact with the stationary phase, and the non-target protein would be eluted off.
Very good. Excellent. And so if we go specifically pH 5 to 5.5, that means all of your other proteins will be positively charged, because below the isoelectric point, your protein will be mostly positively charged. Above the isoelectric point will be negatively charged. So around 5 or 5.5, those proteins you don't care for will be positively charged, and that one of interest will be negatively charged. So because of that, you can use anion exchange. Your positively charged proteins will not interact, they will elute, and your negatively charged protein will interact with the column. Group 1, do you have any questions on that? Okay, good. So continue group 2, how do you elute your protein? That means how do you make it leave the column?
So we said at that point we would gradually change the pH, or you could also do ionic strength of the solution of your mobile phase until it liquids off.
Okay, so you can increase the salt. So what happens when you increase the salt concentration? How would the protein comes off?
Shield the charges.
Yes, so the negatively charged part of the salt, the negatively charged ions, will interact with the column, displacing your protein. So you do that, and then in which direction would you change the pH? Do you increase it or decrease it?
If you decrease it below 4.5, you're going to change the charge on the protein to move it off.
Very good. But because there is this PI of 4.5, you don't want to decrease the pH gradually. You might precipitate the protein. So you want to avoid the isoelectric point. In this case, it's safer to increase the pH, because if you increase the pH, you will have more OH groups, OH minuses from the buffer, and these will displace your protein and will react with the positively charged groups on the column. They might potentially also deprotonate your column, and if the protons is removed, then it's no longer a cation, no longer positively charged, so it's no longer interacting with your protein. So it's safer to increase the pH in this case because you want to avoid the isoelectric point of your protein. Okay, so the last one, maybe one group describes SDS and the other group describes IEF. So Nina or Katie, or do you want to describe?
Sure. SDS page, you have everything be the same charge, so everything's negative.
How do you make that?
You denature by breaking the disulfide bonds.
What did you guys discuss, or you did not discuss that one yet? Yeah. So what did you say? What does you put SDS? What does the SDS do in your sample? Huh? Yeah, go.
It separates it based on size because it has that gradient from the 4% acrylamide.
No. What does the SDS specifically do? The detergent.
It breaks the non-covalent bonds.
Huh?
It breaks the non-covalent
Non-covalent bonds. And then what does it do? Add a negative charge on your protein. So your protein now migrates based on size only because the SDS denatured the protein and imparted a negative charge on the surface of the protein. Okay, so all of them will migrate down based on their size difference, with the smallest protein migrating the most. Okay, so do you guys want to explain IEF? Lisa, do you want to go?
Sure. So in IEF, there's just a charge gradient, and the proteins will just migrate to their isoelectric point and just like focus there, so they'll get a really narrow band.
Yeah.
And you do that first before you do SDS. That way you're not like denaturing the proteins to begin with.
Yeah. So you don't have to run SDS gel unless you want to do a two-dimensional electrophoresis. Then you run your strip on the SDS gel. But yeah, you're right. So you produce a gradient using ampholyte. So it's important to mention that you are producing a pH gradient using ampholytes that are molecules of lots of charges, polymers with different charges. So they will migrate after you run voltage, your current. And then form a pH gradient, and then your protein, once you run the current, you inject your proteins, and once you run the current, they will migrate and stop at their isoelectric point. They will precipitate at the isoelectric. All right, good. I have 7 minutes, 8 minutes. I can start the topic. And we can continue next time. right. So now that we have talked about the structure of the protein, and then we also talked about ways to characterize the structure of the protein, now we're going to talk about how the structure of protein changes upon processing, and focusing a little bit on denaturation, because that's the first change that may occur upon processing. And in denaturation, it doesn't have to always be a negative thing. Sometimes it's desirable to change the structure of the protein or to denature the protein to impart some functional contributions that are desirable. Okay, so we are going to spend probably 2 lecture times talking about protein denaturation and functionality. Okay. So you don't have to know this, but maybe from what we talked about earlier, you can, or you remember a little bit. Which of the following is true? You just have to remember the difference between enthalpy and entropy, and what we talked about in probably a couple of lectures ago. You know? C, yes. So a decrease in enthalpy results actually in an increase in entropy. Enthalpy is the contained energy within bonds, and entropy is the tendency for chaos and disorder. So they are oppositely related. And breaking of few bonds result in an increase in entropy. So it's a chain reaction. It starts with few bonds. It can start with few hydrogen bonds, 2 to 3 hydrogen bonds, can break and result in other bonding, breaking, and unfolding of the protein. A decrease in enthalpy results in a substantial increase in entropy, and that is correct. A slight decrease might result in a lot of energy, free energy, that can build up and cause entropy to increase. Okay, we're going to talk about all of these. So again, you don't have to know this, but can you guess? Denaturation. From what logic, if you have a dry protein powder versus a moist protein solution or paste? None of the above.
A and B.
All of the above. Katie, tell me your thought process.
Well, I was thinking if you're cooking, and you're wanting to brown meat with lard, you don't want it to be wet, and sugar would increase it. But I don't know what's up.
Yeah, that's true. The presence of sugar will increase the Maillard reaction, but we're talking just about the denaturation of the protein itself. So there are other reactions that can happen. So basically we'll talk about, you don't have to know the correct answer, so it's fine. But we're going to talk about the impact of moisture on denaturation, impact of sugar on reducing denaturation rate, and salt, depending on the type of salt, it can enhance denaturation or it can reduce denaturation or reduce the rate of denaturation depending on the type of salt. There are different types of salt, their interaction with the protein would be different. So, can be is correct, but not necessarily must. Okay, when we talk about functional properties, it encompasses a lot of different properties. So it can be organoleptic based on sensation. Looking at the color, smelling and tasting, changes in the texture, the mouthfeel. It could be also touch, smooth, gritty, or turbidity, smoothness, grittiness, turbidity. It can be hydration properties, and this encompasses a lot of different functionality. So hydration, that means interaction with water, can relate to solubility, dispersibility, wettability, swelling, thickening, gelling. All of these different properties has to do with the presence of water and interaction with proteins and water. Surface is what happens at the interface of two different phases. So that has to be with emulsification, foaming, aeration, whipping, any protein-lipid interaction, even flavor and protein interaction that flavor can bind on the surface of the protein as well. Structural, by structural and rheology we mean elasticity, cohesion, chewiness, viscosity, aggregation, all of these different terms. And then there are other less important. So this table really kind of summarizes the categories or the general properties and then the specific functionality related to that. So it's a good studying table. And then next time, we're going to go and talk more about the interaction with water versus the surface activity and then go from there. Okay.